you see the CFTR project appointments. There is a list there of available dates and times, and you just need to tell me what group number you are, what date and time you're looking for. It's kind of a first come, first served basis. And I will update that list reasonably quickly as I get more announcement, or I'm sorry, get more appointments set up.
And if any of your groups are incomplete, let me know, because we have to flesh out the groups, make sure everybody is included in a group, and that the groups are composed of 3 each. Okay. So what I'd like to do is pick up where we left off last time. You should be able to see areas of greatest interest. You see that at the top? Yes or no?
Yes.
You can see it, okay. So just to remind you what we did last time, we spent our time talking about answers to questions that you submitted, topics or areas that you wanted to see more fully discussed, things you didn't understand. So these boil down to a few different areas: energy, enzymes, cell division, Mendelian inheritance, and photosynthesis, with emphasis on oxidation and reduction. We got through the Mendelian inheritance last time, so we're going to start out today with photosynthesis, and using this same set of figures. But before we get there, I'd like to go back to a point about spermatogenesis and oogenesis. And just to remind you, you can see this. Spermatogenesis here on the left, you start out with a spermatogonium, a diploid cell. You go through meiosis 1 and meiosis 2, and you end up with 4 functional sperm cells from 1 progenitor cell. Now, if we compare that to oogenesis, you start with an oogonium, again, a diploid cell, and it goes through meiosis 1 and meiosis 2, and you end up with 1 egg and polar bodies. So the question arises, why are there polar bodies? Why don't we end up with 4 viable eggs? So I'm not sure I have a complete answer for that, but I did look into it. And you remember this came from the special project that, that you did, where on page 5 of that, there were a number of questions asked that were sort of opaque. And so I've looked into why are there polar bodies, and I hope this illustration will help. Let's start on the left here. So the short answer I've given right at the top, you avoid twins because of the polar bodies. So if we look at the beginning in the oogonium, which is diploid, 2N, females are born with many of these, thousands of them, all diploid. So they grow into large numbers. And then once the female reaches puberty, she starts to release one of these every month. So this diploid primary oocyte, one of them comes out of the ovary every month and goes through meiosis 1. And meiosis 1, you remember, you start with one diploid cell and you end up with 2 haploids. But in this case, you end up, the 2 haploids that you end up with, one is a normal cell and the other is a polar body. Polar body cannot be fertilized, and it's at this point that fertilization occurs. If sperm are available and fertilization occurs with this product of meiosis 1, then meiosis 2 starts. And if there is no fertilization, meiosis 2 does not start, and the egg eventually goes away. So then you have meiosis 2 in both the polar bodies and in the secondary oocyte. Polar bodies just generate 2 more polar bodies. The secondary oocyte, which is now fertilized, makes a mature egg and another polar body, which degenerates along with the others. Then after, at this point, this secondary oocyte now has 2 nuclei, both haploid, one from the female and one from the male. And at this point, they fuse to form a zygote. Now we're back to diploid. So we can go back and ask, what would happen if there weren't polar bodies? Well, that would mean that this polar body after meiosis 1 would be a regular secondary oocyte, and it could be fertilized as well. So then you would have twins. I've summarized the steps here in the flow diagram at the right to try to clarify what goes on here. I think this is really interesting, what a difference there is in meiosis between human males and human females. So to start with, girls are born with thousands of primary oocytes. That's these guys, diploid primary oocytes. Then when a girl reaches puberty, she starts to release one of these primary oocytes every month. And it's a diploid, but upon release, it goes through meiosis 1. And that produces a secondary oocyte, that's this down here, plus a polar body. One polar body, and I've crossed it out because it doesn't go anywhere. And after this secondary oocyte is formed, if sperm are available, fertilization occurs. So now you have a cell with 2 nuclei, one of which is already gone through both meiotic divisions. The other one still has meiosis 2 to go. So it does that, and it makes the so-called ovum, the egg, which is still haploid, and another polar body, which degenerates. And now the nuclei, these 2 nuclei, are both haploid, and they fuse to become diploid, and that's the zygote. So is that clear to everyone, how the process works, how it's different between males and females, and why the polar bodies, this is one explanation for why they degenerate, because if they didn't, you'd have multiple fertilizations.
So then are twins a failure of the degeneration of the polar bodies?
It can be. It's my understanding that occasionally a polar body doesn't degenerate. In other words, what happens is the cytoplasm divides unequally. And one of the cells gets most of it, and that becomes a secondary oocyte. The other cell gets very little cytoplasm. It's just a nucleus. And so it really doesn't have the nutrients to go forward. Now, if that doesn't happen, it can be a secondary oocyte and get fertilized as well and can lead to twins. I mean, that's not the only way you get twins, but it's a possibility.
Gotcha. Thanks.
Yeah. So sometimes we have twins. They are the same. They exactly the same. Sometimes they are completely different.
Right. In the case of fraternal twins, if they're completely different, means it can mean that two different eggs or two different primary oocytes are released, and you get the two going through these steps simultaneously. So that's one way to get twins. Another possible way is if the secondary, if the polar body is really a secondary oocyte. So you end up with two secondary oocytes from the same progenitor cell. And
So polar bodies, sometimes they are active, sometimes not.
most of the time not. The normal sequence is polar bodies develop and degenerate. Another way to get identical twins is after the zygote forms, something can happen that causes the zygote to divide into two cells, and each cell gets a full complement of chromosomes. And so then, since both cells are derived from the same nucleus, you have twins, you have identical twins. Okay, and this is all to explain to you why we get such a dichotomy here, where with males, meiosis is normal, and you end up with 4 products of meiosis, 4 viable sperm. In the case of females, from one progenitor cell, you go through meiosis 1, meiosis 2, and you end up with only 1 egg. Okay, if that's clear, then we ended up with Mendelian inheritance the last time. And so we'll start with photosynthesis now. So a couple of you asked, are any animals capable of photosynthesis? And I think the answer is not really, but they can act like it. And here are 2 examples I found. One is a sea slug. See a picture of it here, sitting on a bed of algae. So this slug, its food is the algae. And the algae, of course, are photosynthetic. They have chloroplasts, but the slug incorporates the chloroplasts from the algae into its own cells. And these chloroplasts can photosynthesize and provide food for the slug. And the slug can actually survive without eating any other food for up to 9 months. So that's one type of false photosynthetic activity. Another is this spotted salamander, which again, it has a symbiotic relationship with algal cells. And the algal chloroplasts are in the salamander cells near its mitochondria. So when it's in the light, the chloroplasts produce sugar and oxygen, which the salamander uses. These chloroplasts persist in the salamander throughout its life cycle, which is interesting, and in fact can be passed on from generation to generation, which means that they seem to be able to go through meiosis. And it's from what I read, it's not really clear, but this salamander does better in the light than in darkness. In darkness, it can still eat, doesn't need to be photosynthetic, but it seems to have a better quality of life if it has at least part of its day in the light. So in answer to your question, animals really don't have normal photosynthesis, but they can take advantage of photosynthetic cells. Okay, so let's start with photosynthesis. Where in a plant does photosynthesis occur? It can occur in any part of the plant that's green. If a plant part is green, it means it has chlorophyll, and therefore it can undergo photosynthesis. So for example, a stem, most plants have green stems, so they're photosynthetic, but most of the photosynthesis goes on in leaves. So you see here a leaf, and then a cross-section of a leaf showing the different types of cell, epidermis, stomates, and the bulk of the cells in a leaf are mesophyll cells. And if you look inside a mesophyll cell, you see the nucleus. Remember, plants have very large vacuoles for a number of reasons, including to hold water, and they have chloroplasts. Then if we go further and look inside a chloroplast, what do we see? See an outer membrane, an inner membrane, stroma, and grana. Okay, what are stroma and grana? Grana are stacks of thylakoids. And what is a thylakoid? It's a membrane-bound vesicle. So grana is the plural. Well, one of these is a singular called a granum. And they're simply, as I said, membrane-bound vesicles that are stacked one on top of the other. And we'll talk about, a number of you asked about what's the difference between light reactions and dark reactions. Well, these grana are involved in the light reaction. Stroma are spaces between the grana. And the plural of stroma is stromata. So the spaces, the gray areas here shown in this diagram are called the stroma. And as I said, they're in between these stacks of membranes called grana. And it's in the stroma that the dark reaction occurs. This is where the Calvin cycle, which we'll talk about later, occurs. So we have within a chloroplast, within a single chloroplast, we have both the light reaction and the dark reaction. And we'll talk about the electron transport chain and chemiosmosis. These occur in the thylakoid membrane. So that's the membrane that bounds the vesicle, which is called a thylakoid. And we'll focus in on that membrane a little later.
Excuse me, Professor. One of our question was, does it happens the photosynthesis in everywhere in the cell? It was false because it's only specific for chloroplast, not for every structure of the.
That's right. Photosynthesis occurs only in chloroplasts, and that's because that's the only place that chlorophyll is.
Thank you.
Any part of the plant that is not green, that does not have chlorophyll, does not have photosynthesis.
Thank you.
All right, so let's focus in a little further now. Try to make this bigger. Stages in photosynthesis. Well, at a gross level, right, if you're looking here at a chloroplast and you have the light reactions on the left, the dark reactions on the right, what goes into a chloroplast are water and carbon dioxide, and what comes out is oxygen and sugar, usually glucose. So to repeat, the light-dependent reactions go in the thylakoids. In the grana, these stacks of membranes are really stacks of thylakoids. And that's when the sunlight comes in, it's intercepted within the thylakoids. And we'll go into the detail of what happens in a thylakoid in a little bit. So what comes out of the light reaction is some ATP, but then electron carriers, NADPH. And both of these go into the Calvin cycle, which is the main part of the dark reaction or light independent reactions. So the Calvin cycle takes energy from the light reactions plus carbon dioxide and incorporates the carbon from carbon dioxide, which is a gas, into a solid. And the solid molecules then are metabolized by the use of energy. For example, here 6 ATPs are used, here 6 NADPHs are used, in order to come up with a 3-carbon sugar that can then be stitched together to make, I said sugar, it's not a sugar yet. When it's put together, it becomes a 6-carbon sugar. So that's an overview of photosynthesis. And before going into more depth on that, there were a number of questions like the ones I've posed here. What is oxidation reduction? What is an electron transport chain? What is chemiosmosis? And what do any of them or all of them have to do with ATP production? How do we get ATP out of these processes? So I'm going to go back and remind you of some of the things that we've talked about before. What is oxidation reduction? It's always a pair of reactions. So if you have a reduced substance, A, which has an extra electron, it can transfer that electron to an oxidized substance so that the receptor is now reduced and the provider is now oxidized. So you may remember Scott talks about oil rig. Oxidized is lost, it loses an electron. Reduced is gain. So this is a fundamental reaction that is mediated very often by what are called electron carriers. So if you have a reduced substance, say an organic compound like glucose, it can hand off electrons to a carrier, in this case, NAD, which produces NADH and results in an oxidized substance. NADH then can hand its electrons off to another carrier, for example, a cytochrome, which then becomes reduced. So this is one set of redox reactions. And this goes, there are many of these. So if we look at the electron transport chain, which occurs within a membrane, and there are these proteins within the membrane where electrons are transferred. So think of it this way, that within every one of these proteins, you're getting electron carriers handing off electrons one to the other. So they're going from one electron carrier to the next, from NADH here to a flavin, to something else, to cytochrome. And finally, and while that's happening, protons are being produced on one side of the membranes. So electrons are flowing through the membrane, protons are accumulating on one side, and then at some point, chemiosmosis occurs. So this blue structure represents a number of proteins, including ATP synthase, which takes ADP and inorganic phosphate and makes ATP. So is that clear, the relationship between redox and electron carriers? And a whole series of these occurs in the membrane, and it causes charge separation across the membrane so that chemiosmosis can occur. It also occurs in the membrane. And this is a closer up look at chemiosmosis. It shows you the same thing, electrons from NADH going into the membrane, pass from one carrier to another through the membrane. And while the electrons are doing that, protons are accumulating on one side in the intermembrane space. And when they are passed through this ATP synthase complex, ATP is generated from ADP plus PI. So I'd like to now try to show you this video, which illustrates what I've been talking about. Oh, can you see the video on my screen?
We're still seeing the Word document.
Okay, so I'm going to have to close that and open the other.
After glycolysis, the link reaction and the Krebs cycle have taken place, the reduced form of the electron carriers, that is NADH and FADH2, are sources of electrons for the electron transport chain. These are transported to the inner membrane of the mitochondrion and to the cristae. In the inner membrane, there is a series of electron carriers represented by the white circles here. On the inner membrane, NADH is first oxidized to NAD+, releasing 2 electrons, represented by E- here. Those electrons are picked up by the first electron carrier in the chain. Those electrons are then passed down a series of electron carriers found in the inner membrane, each successive carrier having a slightly higher electronegativity, or that is, a stronger attraction for the electrons. As this happens, energy is released, and this energy is used to pump protons or hydrogen ions via active transport into the intermembrane space. FADH2 enters the electron transport chain at a lower energy level than did NADH. The FADH2 is also oxidized into FAD, releasing electrons which are picked up by the electron carriers in the chain. These electrons continue to pass down the series of electron carriers, and with each successive carrier release energy. This energy continues to be used to pump protons or hydrogen ions from the mitochondrial matrix into the intermembrane space. At the very end of the chain, the final electron acceptor is oxygen. Two de-energized electrons combine with two hydrogen ions and the oxygen in order to form water, one of the products of aerobic respiration. As these electrons continue to be passed down the electron transport chain, hydrogen ions or protons continue to be actively transported from the mitochondrial matrix to the intermembrane space, creating a proton gradient, where the concentration of protons is higher in the intermembrane space than it is in the matrix. Therefore, the hydrogen ions diffuse passively through a channel in ATP synthase back into the mitochondrial matrix from an area of high concentration to an area of low concentration. The enzyme harnesses the available energy, therefore allowing for the phosphorylation of ADP into ATP. This process is known as chemiosmosis. For every 1 molecule of glucose, a total of 32 ATP are produced through the electron transport chain and chemiosmosis. So as a review of aerobic respiration, if you recall in glycolysis, we used 2 ATP, but then produced 4. So there's a net gain in ATP of 2 ATP. The Krebs cycle produced 2 ATP, and the electron transport chain and chemiosmosis produced 32 ATP, and this is all for 1 molecule of glucose. Therefore, in total, for 1 molecule of glucose in aerobic respiration, we produce 36 ATP. So overall, aerobic respiration can be represented in the equation, which would be C6H12O6, otherwise known as glucose, plus 6O2 produces 6 CO2 and 6 H2O, along with energy in the form of ATP, or lost as heat.
How do you see chemiosmosis on the left?
We're still seeing your web browser.
Still, wait, are you still seeing the video?
Yeah, your web browser. So yeah, the video page.
Okay.
There we go.
Okay. All right, so we've just watched this video. So to review, what the video is showing is what these still pictures are trying to show. The transfer of electrons through the series of electron carriers within the membrane. And as the electrons are passed, they pump protons from one side of the membrane to the other. And that makes a proton gradient, a higher concentration on one side than on the other. So these protons then can passively diffuse through a pore in the membrane, which involves this enzyme complex, ATP synthase, and that's what makes ATP. So we're trying to connect the dots here between oxidation and reduction, electron carriers, and ATP production. So let's look at the flow of electrons in the light reactions. There are 2 photosystems called photosystem 1 and photosystem 2. They actually operate in reverse order. Photosystem 2 comes first. Within photosystem 2, there is chlorophyll that absorbs at a certain wavelength, 680 nanometers, and that chlorophyll can absorb a photon of sunlight. When it does, water is split into oxygen and hydrogen. The oxygen goes off as 1/2 an O2. It can later become an oxygen molecule, and 2 protons. The electrons that are released are highly energized by this photon of light to a high energetic state. And this is where they enter the electron transport chain. This is now in the chloroplast. The video showed you what happens in a mitochondrion, where it's very similar to what happens in the chloroplast. The electron transport chain has these electron carriers, which decrease, the electrons decrease in energy as they go through, and so there's a certain amount of ATP that is produced in the electron transport chain in the thylakoid membrane. But now the electrons have lost most of their energy, and they enter photosystem 1, which has a different type of chlorophyll, absorbs at 700 nanometers. It can also receive a photon of light, which energizes it so that it can be passed on to flavins, more electron carriers, which end up generating more reducing power, more ADPH and protons. And these then, the ATP and the protons, go into the Calvin cycle. Remember, the Calvin cycle is unique to chloroplasts, and it is the dark reaction of photosynthesis. This shows you a different view of what I've just said. You have the thylakoid membrane here. You have the proteins embedded in it, which through which electrons pass, being handed back and forth by electron carriers. Light from the sun enters both photosystem 2 and photosystem 1, and out comes energy for the Calvin cycle. The Calvin cycle, remember, is going to make sugar. So this indicates the thylakoid compartment inside the membrane. This is the membrane, and then outside the thylakoid is the stroma. That's where the Calvin cycle occurs, in the stroma. A reiteration, photosystem 2, photosystem 1, the electrons passing from one carrier to another within the membrane, pumping protons to one side of the membrane so that at some point they can go down their gradient through ATP synthase to produce ATP. And this is chemiosmosis. And as you remember, showed you this before, chemiosmosis is nothing but the passage down a gradient of protons or ions of any kind from one side to the other through a channel, generating ATP as they go. And so what is this Calvin cycle? Here's a more detailed look at it. Now, the Calvin cycle is pretty amazing because what it does is takes energy from the light reaction. Remember, the Calvin cycle is the dark reaction of photosynthesis. It takes energy from the light reaction plus carbon dioxide, a gas, and converts that the carbon from carbon dioxide into sugar. And that's done by this amazing enzyme called Rubisco. And it's called carbon fixation, which is the process by which plants convert atmospheric carbon from the gas carbon dioxide to form solid organic compounds, for example, glucose. And the enzyme that does that is called Rubisco. It catalyzes the first step of carbon fixation. And I've showed you the tertiary structure of Rubisco here, just to make the point that carbon dioxide, the gas carbon dioxide, actually binds to this enzyme, and subsequent reactions then connect carbons from carbon dioxide to each other to make sugar. So you don't have to remember the names of these intermediates. What you do need to remember is carbon fixation, that you go from carbon in the form of a gas to carbon in the form of a solid. That happens in the Calvin cycle, and out of it comes glyceraldehyde 3-phosphate, which is a 3-carbon molecule. If 2 of those are put together, you get a 6-carbon molecule, which is glucose. This is the structure of glyceraldehyde 3-phosphate. So 1 turn of this cycle costs 9 ATPs plus 6 electrons. And so you can see that instead of ATP coming out, ATP is going in. 6 ATPs go in to convert this step. And then you have NADPH giving up its electrons for this next step. So is that clear to everyone? The connection between the light reaction and the dark reaction. Light reaction produces ATP and reducing power. The dark reaction converts that along with carbon dioxide into sugar. And that's the source of all the food that we eat and that every other organism uses. Now, we've talked earlier, and there were a number of questions that you had about respiration, and that's because we've talked about respiration as it relates to photosynthesis. So we can take a look at respiration. We're going to have another slide set on respiration, which will have a certain amount of redundancy to this, but respiration occurs in the mitochondrion, and respiration is aerobic. So what happens in respiration, you have a mitochondrion in the cytoplasm. Glucose comes from the Calvin cycle, and a process known as glycolysis occurs to convert glucose, a 6-carbon sugar, into pyruvate, which is a 3-carbon molecule. And when that happens, there is a substrate level phosphorylation that occurs. So you get 1 or 2 ATPs from glycolysis. The pyruvate then is capable of going from the cytoplasm through the mitochondrial membrane, where it enters the Krebs cycle. And electrons from glycolysis can enter the electron transport chain and oxidative phosphorylation. Now, the Krebs cycle can also undergo substrate level phosphorylation. So you get a small amount of ATP from the Krebs cycle. But it's the electron transport chain and oxidative phosphorylation plus chemiosmosis that gives the bulk of the ATP that you get from glucose. I'd like to go through the steps of that. We talk a lot about glycolysis. What is it? You take a glucose molecule and convert it to 2 pyruvate molecules, so a 6-carbon molecule to 2 3-carbon molecules. And what you get from that are 2 ATPs and 2 NADHs, which then can be used in the mitochondrion. Now, this glycolysis is usually shown as one step from glucose to pyruvate, but in fact, it involves 9 steps shown here. So you don't need to learn these, but I just want you to be aware that, that glycolysis is a very complicated process with 9 steps in it. It occurs in the cytoplasm. It's not just a one-step process to go from glucose to pyruvate. Any questions? Are you following me, or am I going too fast? Slow me down if I am. Okay, let's then look at the Krebs cycle. The Krebs cycle occurs in the mitochondrion. So here you see a cell. This big blue thing is the nucleus, and the little structure is the mitochondrion. There you see a blow-up of mitochondrion, and then if you look at just one portion of the membrane of this mitochondrion, that's what you see in this picture. So again, we start with glucose. Remember, where did the glucose come from?
Food.
The Calvin cycle?
Right, comes from the Calvin cycle. And so we have the glucose, which can yield, when it's converted to pyruvate at substrate level phosphorylation. The pyruvate is then capable of passing across the mitochondrial membrane along with the electrons that are generated from glycolysis, so that the pyruvate, once it's inside and it's in the matrix of the mitochondrion, it can get converted to acetyl-CoA. And acetyl-CoA is the first step in the Krebs cycle, also known as the TCA, tricarboxylic acid cycle. And when the Krebs cycle turns, it also can generate substrate level phosphorylation to produce a small amount of ATP. But the main thing the Krebs cycle does is produce NADH and FADH to go into the electron transport chain, which is not in the matrix, it's in the membrane of the mitochondrion. So again, here we have what we've been talking about and what the video showed, these proteins in the membrane that represent a series of electron transport steps, a series of carrier reactions where electrons are handed off from one to the other. The electrons pass through the chain, and as they do, they're pumping hydrogen protons into this intermembrane space. And when the concentration of protons gets high, chemiosmosis occurs. So these 3 purple objects are where chemiosmosis happens. It's where the ATP synthase is. So ADP plus inorganic phosphate is converted to ATP from the energy of protons passing across the membrane. Electron transport and proton pumping is what produces ATP. Now, there was a couple of questions about how is acetyl-CoA involved in an anabolic process. I'll show you that in a minute. But what we're concerned about mainly here is that acetyl-CoA initiates the Krebs cycle. So any question about this?
Yeah, I got a question. Since we're going over a few cycles, are you going to be specifying which ones we're going to know more in depth about? Or should we just know broad like details of each one?
They're really just two cycles, the Kelvin cycle and the Krebs cycle. You need to know where each one occurs. One is in the chloroplast, the other in the mitochondrion, and what each of them does. The Kelvin cycle takes energy from photosynthesis, from the light reaction, and converts it into sugar, food. The Krebs cycle takes that food, glucose, and converts it into ATP.
Sounds good. Thank you so much.
Any other questions?
The Krebs cycles, teacher, it's related to animals and plants both, but Calvin cycles, especially for plants.
Yes, that's a point that should be emphasized. The Kelvin cycle occurs only in plants. The Krebs cycle occurs in both plants and animals. In other words, you don't. The Krebs cycle is aerobic respiration. It's converting glucose to energy that our muscles need, or our whole body needs in order to function, needs ATP. And that ATP comes from the Krebs cycle in both plants and animals. So it's an important distinction. The Kelvin cycle occurs only in plants. The Krebs cycle occurs in both plants and animals.
Thank you.
It's a good question to ask on a test. All right.
The Krebs cycle, you said it occurs only in the matrix of the mitochondria?
Yeah.
Okay.
It shows it here. The TCA cycle is in the matrix.
And that's separate from the electron transport chain?
Yes, that's another important point. The electron transport chain does not occur in the Krebs cycle. It occurs in the membrane. So the Krebs cycle is in the matrix. The electron transport chain occurs in the membrane of the mitochondrion, the inner membrane. But it feeds off of the reducing power, the NADH and FADH, that are generated by the TCA cycle, Krebs cycle. These electrons go into the membrane and are passed from one carrier to another within the membrane. And as they do that, protons are pumped into the intermembrane space, which causes a gradient, high concentration in this space, much lower concentration in the matrix. And so when these protons are pumped out of the membrane, they go through this enzyme called ATP synthase, and that's what converts ADP to ATP. That's where energy is produced from these protons that are produced from these electrons that come from the Krebs cycle. So it's a sort of a long series of steps, starting with a photon of light in the chloroplast to generate energy for the Calvin cycle. The Calvin cycle fixes carbon into glucose. The glucose is then used by the Krebs cycle to generate a reducing power, which can then be converted by the inner mitochondrial membrane into ATP. Anything else? A couple of you asked about acetyl-CoA. What does it do? It can be involved in both catabolic and anabolic processes. So in the Krebs cycle, ATP is generated as a result of the Krebs cycle. And acetyl-CoA is the first molecule. It's a 2-carbon molecule that is the first step in the Krebs cycle. Again, you don't have to know all these intermediates, but you do need to remember that the Krebs cycle generates energy for the electrons for the electron transport chain, and it has a substrate level phosphorylation.
So we don't have to know how many ATP from each cycle it's happened from Calvin or for Krebs.
Yeah, you should be able to know the stoichiometry. I don't know if I have it here. I guess not. But if you look in the previous slide set, it shows the stoichiometry of ATP generation, where we talk about the different types of making different ways to make ATP. There's substrate level, there's oxidative phosphorylation, and there's photophosphorylation. Well, photophosphorylation only occurs in the chloroplast, but for generating energy in animals don't have chloroplasts, substrate level phosphorylation and oxidative phosphorylation are the main ways that ATP is generated. Substrate level phosphorylation is relatively inefficient. For each molecule that's processed, you get 1 or 2 molecules of ATP. Whereas in oxidative phosphorylation, and I don't have the slide in this set, but it's in, as I said, in the previous set that you can find in Canvas, it shows where the different, it counts up how many different ATPs you get from substrate level phosphorylation and how many you get from oxidative phosphorylation. And it adds up to either 38 or 36 ATPs from 1 molecule of glucose in oxidative phosphorylation. And the difference in numbers of ATPs depends on how you count. In one case, you count ATPs that are not part of the net ATPs, and in the other case, you don't count those. So go back and look at that previous slide set, and you can see the story.
Yeah. So then it will be like all the ATP that we get from Kelvin minus the dark. When it's dark cycle, we will spend ATP, so we will minus that, right?
The dark cycle.
It's take ATP, so it's will take energy.
But wait, back up. This ATP that the dark cycle, Kelvin cycle uses, that's not generated from the mitochondria.
No, it's generated from the light. The light. So they take it. That's what I'm saying. We will minus what we get from the light, and then minus that from we spend it in the dark.
No, when you're counting ATPs start with glucose, which is generated by the Calvin cycle. You start with glucose and you ask how many ATPs do you get from one molecule of glucose? And so there are two substrate level phosphorylations, and then there's the electron transport chain here.
Yeah.
You get a substrate level phosphorylation here, but you're starting with glucose, which came from the chloroplast. Or came from somewhere, not from the mitochondrion. So the glucose comes from the food you eat, and you get substrate level phosphorylation, and then you get most of the energy from oxidative phosphorylation.
Okay. Thank you.
Okay, back to acetyl-CoA. The importance of it is that it's the first step. It's necessary for the Krebs cycle to operate. It feeds the carbon into the Krebs cycle. And it's considered a crossroad compound because it can be either catabolic or anabolic. So it's catabolic, it's what we've been talking about, extracting energy from carbohydrates, fats, and proteins. It's anabolic in that it can be used to build fatty acids. So this diagram is more complicated than I'd like, but what I'd like to show here is that within the mitochondrion, acetyl-CoA, in addition to the Krebs cycle, it can enter the fatty acid synthesis pathway and make fatty acids. So, yeah, acetyl-CoA, a couple of steps, you get fatty acids represented by these chains. And to remind you, this is what fatty acids are. It's a long carbon chain terminated by a carboxylic acid group. And this long carbon chain can be either saturated or unsaturated. And you need to remember this because of membrane physiology. It's phospholipids that are essential for function of the membranes. And remember, we've just been talking all day about membranes and how all these important things happen in membranes. And membranes are basically fatty acids, phospholipids. Okay, I have another slide set on respiration that there's really not time in this period to go into. So that's where we'll start next time. That slide set is, it has some new information in it, but it also is a review of some of the things that we've talked about today. So that's the end of this class period. Any questions before we go? Make sure that you contact Scott about setting up your cystic fibrosis project review. And don't forget that Scott has a workshop starting at 2 o'clock.
Yes, I do.
So, okay. So if there's nothing else, I'm going to stop sharing. And we will see you on Monday.
Alrighty, see you on Monday. Thanks so much.
Okay.